Okay, thank you for that intro, Sahar. So our project is we're called the fMRIusic team, and our project is determining neural correlates for um, different affective responses to musical stimuli and classifying musical affection through machine learning. I'm Ayan, and my group mates are Oren and Maya.
Our project is mainly focused on analyzing fMRI data. fMRI stands for functional magnetic resonance imaging, and it produces 3D images of the brain. Unlike MRI, which gives anatomical data, fMRI data allows for correlation between the activation of different brain regions and specific tasks, such as listening to music. fMRI utilizes the properties of oxygen in blood to determine the blood oxygen levels across different parts of the brain. When a part of the brain is activated, there is more blood flow to that region. This increased vascularization causes a change in the blood oxygen level dependent signal, which the fMRI machine is able to measure. fMRI data therefore shows which regions of the brain are activated while a person is doing a certain task.
So fMRIprep is an fMRI pipeline that takes raw fMRI data and turns that into a preprocessed form of it. fMRIprep implements tools from all different software packages such as FSL, ANTS, Resurfer, and AFNI. It outputs preprocessed quality reports so we can identify the outliers and remove them. It automates and parallelizes processing steps. So basically that means that it just helps it run faster. Moving on to XCPD. XCPD is a post-processing pipeline that picks up right where fMRIprep left at, and it generates the noise bold images, parcellated time series, and FC matrices. It works in BIDS and IPRES framework, and this is great for quality control because it gives quality reports, and again, we can identify the outliers and remove them. So far, XCPD has worked way better than CPAC and FSL in the post-processing state after the fMRIprep outputs.
So FSL is a software tool which we use for high-quality model-based fMRI data analysis. It has an easy-to-use graphical user interface, which makes it helpful for efficient processing of large amounts of data. FEAT, which is the analysis portion of FSL, automates as many of the analysis decisions as possible and allows for easy, robust, efficient, and valid analysis of all our subjects. FEAT gives us enough flexibility to also allow for sophisticated analysis of the most intricate subjects that give us difficulty. So we ran FEAT first level analysis on almost all of our subjects. So our project focuses on targeting the brain's auditory network. So the brain's auditory network works via auditory messages that are conveyed to the brain. The auditory network is located on the superior portion of the temporal lobe of each of the brain's hemispheres. The auditory network is interconnected. Any damage to the auditory cortex can lead to a loss of sound for a person. And the primary auditory pathway exclusively carries auditory messages throughout the brain, while the non-primary pathway carries all types of sensory messages.
So for our data set, we had a total of 40 subjects, and each of these 40 subjects listened to 4 recordings of different clips. And these clips were a happy clip, a short sad clip, a long sad clip, and a rest clip. And we took the study from online from the University of Columbia. And basically, it explores the brain activity in 39 subjects listening to music. 1 subject was ruled out based on it was just an outlier. Brain activity, we analyzed it from playing happy and sad audio, and we converted the functional data and uploaded it through the ASDRP server. The functional data basically just measures neuron activity, while the structural measures anatomical structure.
So here are some examples of fMRIprep outputs, which we use for preprocessing. So on the top left, we can see a heat map, which summarizes the correlation structure among confounding variables, or variables that could explain activity that isn't related to the task that the subjects are going through. And to the right of the heat map, we see a graph which shows the magnitude of these correlations. On the bottom of the slide, we can see an anatomical preprocessing step. This step is known as spatial normalization. And here the patient's brain is mapped onto a reference or a template of the previously collected brain scans that are then compiled into an average template of the brain.
We began with acquiring our data, From there, we ran the data through a preprocessing pipeline using fMRIprep. We then used XCPD, which is a fMRI post-processing pipeline developed for fMRIprep outputs, which generates bold images and many others. We then did first-level analysis through FSL feet, and this was a harder challenge considering the technology was much older. For machine learning, we took the outputs from XCPD and time series data and used this data to apply feature extraction with PCA and UMAP to reduce the dimensionality, and this allowed us to better visualize and analyze the data. Our team also used KNN, SVM, and CNN, which are different machine learning algorithms. When using FSL feet, we created an analysis model to run preprocessed files. FSL allows for easy analysis as it automates many decisions made during the process and allows for flexibility when working with complex data. It allows us to create an experimental model to run the data on, which will then show which areas of the brain are activated as a response to the stimulus. In this case, listening to different types of music. At the bottom of the slide, there's an example of an output from FSL feet and how it shows the different brain regions that are activated. There's also an example of an experimental model that we used.
So FSL feet, we first ran it on data outputted from XCPD, which is one of our post-processing steps, but the images indicated that brain activation showed no significant activation, which we could identify due to no resting states for comparison. So we then turned to the original fMRIprep process data to run our analysis. So feet allowed us to describe the experimental design for a higher efficient analysis and then create a model that should fit that data. And it would then tell us where the brain was activated as shown by, in response to stimuli as shown in red on the top image. And then on the bottom, we have a time series graph, which shows all the signals that are measured at each voxel of the brain across the entire task taken over time.
As for our results, all 40 of our subjects from our study moved on to post-processing steps after fMRIprep. XCPD was successfully run on 38 of the 40 subjects, but unfortunately 2 of them were removed from analysis. We have also conducted FSL feet first-level analysis on all subjects. The picture on the top right shows the happy, sad, and rest distribution of all of the subjects. And as you can see, the green, which is meant to represent rest, clusters on the left side, whereas everything else seems scattered. And this shows the PCA and UMAP scores. If you use PCA and UMAP, you would only get an accuracy score of around 40%, but just using PCA, we got an accuracy score of 82% when run with SVM and 89% on KNN. Our objective is to continue to employ these machine learning models for the purpose of detecting the activation of specific brain regions and correlating them with emotional responses to musical stimuli.
So in the future, we want to first work on time series analysis, then functional connectivity analysis, and then finally develop a machine learning algorithm. So first, time series analysis is basically the signal that is measured at each voxel across the entire run. And functional connectivity analysis basically shows a conclusive result through functional connectivity data and ROI mass. ROI mass are mass that define a region of interest, and it's used as a mask to remove pixels from an image, whether it's in the region or not. Finally, we will use machine learning algorithms to extract the results on which brain regions of the human brain are active during emotional responses to music. So we'd like to thank the following people: Saharjan Ikia for her guidance and support throughout the research process, ASDRP with providing us the resources to conduct this research, and also the study from online from the University of Columbia and Southern California.